improve this. Okay. We can inject a purpose into these enzymes. That's called a directed evolution. Okay. So this one doesn't work. You have to go after the enzymes that show the promiscuous activity. Okay, that's important. And just to think about just forgive me if I talk too much about chemistry here, okay? But I think this is a good story or good narrative to tell, okay, here. So think about this P450. How do they oxidize substrates, organic compounds? This enzyme contains a cofactor. This is an iron protoporphyrin. It has an iron center, which sits in the middle of the protein, in the center of the protein. There's a hydrophobic pocket. Okay? In the presence of oxygen, it gets converted into this widely famous compound known as compound 1. Okay? Oxo iron 4 complex. This is the reactive species that can activate the CH bond and insert oxygen to create CO bond. What can we do? So, one postdoc, Eric Brastad, he used to be a PhD student with Peter Schultz in Scripps. He joined the Francis Arnold lab. He imagined, can we use these enzymes to create carbon-carbon bond, carbon-nitrogen bond, even carbon-silicon bond? Because these reactions are widely used in synthetic chemistry lab. And people are looking for catalysts that can do this type of reaction. But can we simply just replace oxygen here with carbene or nitrene? Right? Then we can convert this enzyme to, following similar catalytic cycle, we can convert this enzyme to a new catalyst for completely different unnatural reactions. Right? How can we do that? So can we hijack directed evolution? Can we hijack this natural enzyme for new reactions? Remember, as I just mentioned, okay, we need to check whether the natural enzyme, Y type enzyme, can catalyze the reaction of interest, right? And then if with the enzyme, natural enzyme can do that, we can take advantage of directed evolution to further improve the efficacy or efficiency of the process. Right, before we jump into directed evolution, first we need to check whether the natural enzyme can do this. Sorry, the laser is not working well. Yep. They tested, it turned out that actually Pedro, the graduate student, worked in the lab, tested this reaction. It showed some minimal reactivity, the Y-type enzyme just mix, even if you simply mix him with the substrate, it can convert with this diazo acetate, okay? That's one of the starting material that can generate carbene species. Okay, then you can create a carbon-carbon bond. This is a cyclopropane resulting from this reaction. You can do that. Okay? Once you have this reactivity, they applied the directed evolution. Okay, they were able to dramatically improve the efficiency of the reaction. And if we have chemists here, they will be super excited because in some variants, if you just change one residue, this is a big protein, contains around 250 amino acids. If you just flip one amino acid residue, it can change the stereochemistry. It can change the orientation of the resulting material. It's a dream goal for a lot of people who are working on, asymmetrical catalysts. Okay. And but the problem is, it turned out that the enzyme is not stable. Also, it's not compatible. Although they can, with the directed evolution, they can do a lot of turnovers in the test tube, but it's not compatible with living system. The beauty of protein is that in future we can integrate it with living system. We can use cellular machinery or cell as a factory to produce these compounds. But with this enzyme, we cannot do this. Because when you do this type of reaction, you have to add a very strong reducing agent to jumpstart the reaction. Okay, the iron 3 in the heme has to be reduced to iron 2, and then it can be used to catalyze the reaction. Okay, so how can we address this dilemma? Okay, one hand, this protein in test tube can show good reactivity, but on the other hand, if you put them into cells, they don't show this reactivity. How can we accommodate, reconcile this conflict? So this Pedro is clever. He just look into something, think out of the box, look into something nobody else has ever looked into. So there's a ligand, it's called cysteine. There's a sulfur that coordinates with the RNA. Okay, this cysteine residue is highly conserved in P450 protein family. This is a big family. This type of protein exists across all the forms of living systems. It replaced this cysteine with another residue called a serine. They are very similar to each other. You only need to replace sulfur with oxygen. Okay. Conventional wisdom believes that if you remove this cysteine, the protein will not be able to bind to the cofactor because it serves as a coordinating ligand. And also, even if it can still bind, but that doesn't give you natural property of interest. But regardless, they did it. It turned out that, yes, the enzyme changed the color. The original enzyme is red, and then once they replace that, the enzyme becomes green. Okay? And also the most important thing is that with this type of enzyme, you can just express them in E. coli, and then use the whole E. coli as a whole cell as a catalyst. You can also convert the substrate to generate this cyclopropane product. You can do this carbon transfer reaction. What's the magic thing that's going on here? So basically, if you replace sulfur with oxygen, you can dramatically increase the redox potential of the RN center. Okay? Then those moderate reducing agent inside of the cells will be able to reduce RN3 into RN2, and then you can jump start the reaction to generate the carbene and then transfer the carbene to generate cyclopropane. Okay? And then they even did another crazy thing. Okay? They just did a saturation mutagenesis. What's the meaning of that? You can replace cysteine with the other 19 possibilities, other 19 possible amino acids. It turned out that many of these genetic variants also can show unexpected reactivity and stability in living systems. Okay? This is something nobody has thought about this before. Not only that, you can also not only create a carbon-carbon bond, you can also use this enzyme to create a carbon-silicon bond and a carbon-boron bond. Okay? It's carbon transferring. The carbon can be inserted into SiH, silicon hydrogen bond, and it can also be inserted into boron, hydrogen bond. So you can create a variety of unnatural, artificial chemical bonds using this type of enzyme. So this type of enzyme can also be very sophisticated in creating very complicated unnatural compounds. You can convert this alkyne triple bond. You can do double propocyclination. Generate double cyclopropane to create this bicyclic structure. Okay, this paper was also published in Science because this is a completely unnatural reaction, and also you can generate a very complicated product with this type of technology. So people may ask, what did you do in Francis Arnold lab? So far you haven't talked about what you have been able to accomplish. So when I joined the lab, Francis asked me, can you think about something which is different from what other people are doing in the lab? Can we think about out of the box? Okay. Pedro did that. He replaced those cysteine residue, highly conserved in P450 enzyme, replaced that with a variety of other residues, and then which lead to new reactivity. So if we look into protein sequence space, we emphasize in conventional or traditional protein engineering, we emphasize the importance of sequence. Okay? Size. We do truncation, we make the protein more concise, more stable. Okay? And of course, unless you use unnatural amino acids, we don't need to care about stereochemistry because look into all these 20 natural amino acids except glycine. They are all L amino acids. You don't have to worry about the stereochemistry unless you use unnatural amino acids. So traditional protein engineering focuses on these 3 dimensions. Okay, what else can we do? So if you are a polymer scientist, protein is a polymer by nature. Okay? When we talk about the property of a polymer, we always talk about 4 aspects, right? Size, sequence, stereochemistry. What else? Topology. Okay? But sadly, most living biopolymers in living systems, they are linear, right? They are linear. So can we introduce another dimension for protein engineering or directed evolution in general? Can we engineer the topology of protein molecules? Can we have a control over these engineered protein molecules? How can we do this? Again, we are puzzled. We don't know where to go. What shall we do? We look into nature. Okay? We get inspired by nature. So the rationale is that if we can make those nonlinear polymers, we may have access to materials or new molecules with properties that never been explored before, right? So how can we do that? This is another side note, okay? In the past few decades, in all important biotechnology, most of them are derived from our understanding or new knowledge about, biological process in some simple organisms, PCR, CRISPR-Cas9, or think about this channel rhodopsin. This is the protein derived from marine microalgae, okay? So it turned out that the same organism that contributed CRISPR-Cas9 technology, okay? This is called Streptococcus pyogenes. This is gram-positive bacteria, a human pathogen, okay, causes disease. The surface of this protein contains a multi-domain protein called pili, which is believed to be involved in host pathogen interaction. An important feature is that this pili, this protein contains an isopeptide bond, a covalent bond formed between lysine and aspartate residues. Nature is able to engineer protein topology. Think about another molecule inside our body, insulin. This is not a linear molecule. After central dogma, after translation, yes, it's a linear polymer. But there's another process in nature called post-translational modification. Nature was able to encrypt this type of information into the genetic sequence. And then they can control the formation of disulfide bonds, or in this case, isopeptide bonds, between protein polymer chains. Okay. Can we harness this type of phenomenon, post-translational modification, to engineer protein topology? That's the question we want to answer. How can we do this? How can we just take this, put that into another protein, and then control, make nonlinear protein molecules? So another interesting study led by Markow's group in Oxford showed that if you split this pili protein into 2 fragments, the resulting fragments, one is known as spike-tap, the other is called spike-catcher. Okay? If you mix them, you don't need to do too much engineering. If you mix them, they can also form this intermolecular, lead to the formation of isopeptide bond between these molecules. It's an intermolecular process. Basically, in doing so, you can convert an intramolecular reaction into an intermolecular reaction. But for something to be useful, especially in the field of chemistry, okay, we want to be efficient. Not something that can, if you can do it with 10% conversion, that's useless. Okay, you have no control over the process. So how can we improve the efficiency? Okay, so we want to have something that's super efficient, highly selective, and it can rival, be comparable with another type of paradigm in chemistry, click chemistry. Okay, how can we do that? How can we improve the efficiency of such a reaction? So then it requires the utility of another important tool called the phage display to improve protein-protein or protein-peptide interactions. So that's why half of the price of this year went to these two biochemists. They invented this technology called phage display. What phage display is able to do is that it can display one of the reactants or one of the partners on the phage surface, and then you immobilize the other on the column, on solid substrate. Okay? Randomize the genes that you inserted into the phage. Then you can ask these phage to display those genetic variants on their surface, and then you do the capture and enrichment. Then you were able to evolve those new partners that showed higher improved affinity toward those immobilized substrates. Okay? Remember, Manfred envisioned that we need a molecular evolutionary machine to do directed evolution. Phage is a perfect and ideal evolutionary machine because that's a perfect system that integrates phenotype with genotype. You change the DNA sequence, and then you result in the protein molecules with altered sequence, and then that will, the phenotype, the physical property arises from these genetic protein variants. It's a perfect system. You can correlate genetic information with the protein sequence. So this is great technology because I think in 1970s we have had this great revolutionary technology called the hybridoma technology that enables us to mass produce, to engineer, to create monoclonal antibodies targeting different epitome, different molecule substrates you can define, okay? But there is a drawback for hybridoma technology. That's another Nobel Prize winning technology. The problem with hybridoma is that they can only generate mouse monoclonal antibody. If you want to use monoclonal antibody for human therapies, you have to humanize it. It's extremely challenging, okay? That's a big bottleneck for using monoclonal antibodies for therapeutics. So with phage display technology, now you can just display those antibody fragments outside of the phage. Then you don't have to worry about humanization problem, okay? And then eventually, with the maturation of the technology, people developed this. It's known as FAG, okay? Antibody fragments, okay? To treat diseases like arthritis, rheumatoid arthritis, a lot of other, inflammation-related disease. Okay, so you can circumvent humanization problems for developing monoclonal antibody. Okay, so let's go back. Okay, how can we evolve spike catcher to make it click chemistry-like, reaction? So Howard's it's simple, very straightforward. First, Howard's displayed spike tag variants on the phage surface. Okay, he was able to improve the reactivity by mutating spike tag. Once he ended up with several improved mutants, he further displayed spike catcher on the phage surface. He combined both, and then you were able to generate a new spike catcher chemistry that showed improved reactivity in test tube. Okay, once we have these optimized genetic engineered tools, so then we can use them to control protein topologies during cellular synthesis. So that's what I did in Arnold's lab in collaboration with Dave Terrell, another polymer chemist at Caltech. So we were able to make cyclic proteins, cyclic polymers. By it's simple. You begin with genetic design. You have elastin-like protein gene in the middle, and then you put the two spike catcher genes, on both ends, and then you put the gene into E. coli. You are able to make cyclic proteins. You can also you can move one of the reactants into the middle of the gene. You can make catapult-like molecules. Okay, my colleague, Professor Zhang Wenbin in Peking University further demonstrated that with these newly generated protein tools, they can create unconventional protein molecules with unconventional diverse topologies. They can make protein lasso. It can serve as a molecular switch. Okay, change the condition, you can change the conformation. Okay, it's a lasso. They can also make this branched protein molecules, star-like protein molecules. It's a good design strategy for creating synthetic vaccine. You can display multiple antigens on the surface, on these branches. And also they were able to form this physically entangled catenary structure. Okay. And a student in my lab, Zhongguang, is able to use cellular machinery to make 4-arm proteins. Okay. Once you have these 4-arm building blocks, you just ask E. coli to make it, put the gene into the E. coli, and ask the E. coli to make these 4-arm proteins. And then you mix it with proper building block, you can make an entirely protein-based network. Okay? Then this is a hydrogel material. Okay? So with hydrogel, you can do a variety of drug delivery or tissue regeneration. You can just use genetic programming to alter bio and the physical properties of this material system. All right, so this is pretty much I can present today. I just spent the last couple of days looking into literatures and try to put together these pictures. And this is important. I hope what I present today is able to evolve and inspire, even spark some new ideas from the audience, from the young audience. Okay? And more importantly, don't pay attention to what has already been done. Okay? The most important thing is looking for what remains to be done. Okay? This is more important. Look into the future. Directed evolution is a great tool, but it's more a new way of thinking. Okay? How can we harness this type of philosophy? Francis Arnold is a big believer in brute force. Okay? He said that evolution is something gentlemen don't do it, but she's not a gentleman. Okay, you don't have to be gentleman to be a good researcher. Okay, you have to believe in the force that's, has already been perfected by nature. Okay, directed evolution. You can come up with a variety of systems that can be improved, optimized. It's algorithm. It's simple. It's the stupidest algorithm, but that can be very useful, very easy to use. Okay, so let me end up with this lecture with Mary Cull's quote. Okay, be less curious about people. Okay, people expect me to talk more about Francis Arnold. I think more important, we need to be curious about ideas, about science or engineering problems in general. So, all right, thank you very much. If you want to know more about directed evolution, I hope you can join this event. Okay, it's by Hui Min Zhang. They adopted this directed evolution into more complicated biological systems. And also we invited some group alumni for this workshop. Yoling Chong is one of them. He's now doing synthetic biology. Okay, borrow some elements from directed evolution to perfect the biological system for other interesting applications. So any question from the audience?
Thank you for sharing this very wonderful story. So actually I have a question about this termination, about this direct evolution. Because you mentioned about the random mutagenesis, and from this functional screening, and finally you can find the very good enzyme. So I'm wondering, is this called the direct evolution? Because I'm not in the science field.
Evolution is simple, right? You combine, randomization with selection. That's a process called evolution. Why do we call it direct evolution? Because when we perform evolution in laboratory, we have a purpose. We inject a telos or end to this process. Okay, we always tilt the process of evolution toward our own goal. For example, if we are interested in thermostability, you would like to subject those genetic variants to protein mutants. on the, to high temperature, see which one survives. And at the end, once you get the thermostable protein, varied, okay, that's the end goal. Okay, it's called directed evolution. There's a direction, there's a purpose for whatever you're doing in the laboratory.
Thank you. I have another question.
Because we ask about the purpose of natural evolution. Do we know that? We don't know, right? I mean, this is a very philosophical problem. Okay, what's the telos of natural evolution? We know nothing about it. Okay, there's a big debate between who believe in God and who do not. Okay, so I think that's why we call it directed evolution. We inject the purpose, the telos, into the process.
Thanks. I have another question. Because for each optimization process, you have the parent protein, and also the final, the good one, you selected the stuff through this functional screening. So I'm wondering if we compare the parent protein and the final protein, there should be some very insight about the structure-function correlation. I think this is the most important lesson we can learn from this.
Yeah, that's another interesting, you can call it byproduct of the process of directed evolution. It provides a new aspect or perspective for our understanding of how protein molecules work. Okay, and some of these detailed information cannot be revealed by routine traditional biochemical studies or structural studies. You have to do evolution, because it turned out that some genetic mutations that occur to a very remotely, to the catalytic site. Okay, you cannot predict the impact or influence of this type of mutation to the reactivity of the enzyme, but it turned out that through evolution, these sites can be revealed. Okay. Also, sometimes there's a lot of so-called neutral mutations. People believe if you just replace, serine with threonine, you're not going to impact the protein activity dramatically. But that's not the case in some directed evolution process. Neutral mutations can also generate a big impact on the protein activity. Okay, you have to get the molecule. Okay, then you will be able to see that interesting phenomenon. So another thing, I think another important development in the field, represented by David Baker in Washington University, they are perfecting the technology using computational design to design protein molecules, functional proteins or catalysts. But nowadays, at the best they can do is to make some biocatalyst that shows minimal reactivity. Then you still need directed evolution, experimental lists to improve, to optimize the candidate. Okay, so it turned out that we cannot get away with directed evolution. No matter how smart you are, how clever you are, we still need directed evolution to perfect these designs, artificial systems. Right. I think another interesting example is in the field of synthetic biology. People build up a variety of genetic parts. It's called a genetic circuit, or let's call it a bio bricks. Okay, when they try to put together into a cellular system, compatibility is an issue. Okay, although they work well independently, but you put different genetic circuits into one cell, they have compatibility issue. Okay, and then some bioengineers try to harness the power of directed evolution to fine-tune this biological system to address or reconcile the incompatibility problem. So you cannot predict in these prior knowledge, okay? Any more questions? Quiet as my class. The turnout is, the audience size is smaller than my class actually, okay, but it's as quiet as. All right, so I won't keep you here, so thank you for coming. And Hope I can see some of you in my future class. Okay.对。